Hello again. Today, in this video, we're going to reach the third level of understanding viruses. I hope the previous two videos made a slightly smoother transition to this video. I think in the previous video, level 2, things started easy and then got a little bit complicated. And I think reaching level 3 of understanding is going to make it easier to get the full picture. Sometimes understanding something halfway is worse than, or actually is more difficult than understanding it completely, because you get like questions, why are you explaining that? And you're not really reaching the full answer. So I think getting the whole subject at once is a healthy way of understanding it. So in the first video, we talked about viruses as boxes that contain information that contains instructions on how to make these boxes. In the second video, we explained that this box is made of protein, known as a capsid, and inside is a genetic material that is either DNA or RNA that contains instructions on how to build this virus. We also talked about how cells do that, how cells transcribe their DNA into a message RNA, which is a copy that contains instructions of making a specific virus, and then this RNA is translated into a protein. In this video, we're going to try and combine these two things together and try to understand how viruses, such as the coronavirus, behave. So, the coronavirus is an example of a virus that is an RNA virus. And it's called corona, coming from a word that means crown. Because if you look at this virus in a microscope, under a microscope, it's going to look something like this. It's more circular, actually. It has these spiky things like the crown, of a princess. These are known as spike proteins, which are functional proteins that allow for attachment to certain cells, specifically the cells of human lungs and other animals, in fact. So because of these spiky things, we called it the crown virus or the coronavirus. And it's an RNA virus. What does that mean? It means that inside this capsid, the genetic material is made of RNA. Now, what happens when this virus infects a cell? Here's a cell that is a lung cell, and this is the coronavirus. Oh, that's really not a great picture, but let's take it as it is. The spikes are used to attach to the surface of the cell, and then the RNA of this virus gets injected. Now the cell looks at this RNA and says, okay, is this a message? Because RNA usually behaves as a message. Is this message coming out from my nucleus asking me to build a certain protein? The cell cannot figure this out. It just looks at this RNA and assumes it's some sort of a message. So it proceeds to translate it. It translates it and makes out of it protein. Now, this protein is going to be the viral protein, the viral capsid, which includes structural proteins and functional proteins. And among these functional proteins are the spike proteins and others. So we've made the capsid of the protein. Now we want to make copies of this RNA. But wait, there's a problem in here. Let's take a look at the cell. Let's go back into the general picture. We said that viruses contain information, a sheet of instructions that tell you about the materials that should be DIY, do-it-yourself virus. It should contain a set of materials that you can find in your house, right? And the house in this case is the cell. So the cell should contain all of the tools and materials necessary to build the virus. In the case of the coronavirus, which is an RNA virus, we do have the tools to actually build the protein that the virus is made of from the RNA that is inside it. However, we cannot make RNA from RNA. The cell does not contain tools to make RNA from RNA, because what we mentioned before is that the cell normally can make RNA from DNA, and that's the message, and then this RNA is converted to protein, or it can make DNA from DNA in the process of replication, where we make DNA for other cells. But we cannot make RNA from RNA. This is something that the cell does not know how to do. Interestingly enough, this RNA virus says there's no problem. Just go to page 3 of the instructions, or whatever page, and you will find a page that describes how to make tool that makes RNA from RNA. And this tool is a protein. And the protein is something that the cell can make. So these coronaviruses are really smart. They tell the cell how to make a tool that it doesn't even have. In scientific terms, the genetic material of the RNA virus is a long string of information, and it contains regions that code for structural proteins, like the proteins that make up the virus. And then there are regions that contains non-structural proteins, which can be functional. Some of these proteins can be the spikes, and some of these proteins are proteins that are used to copy RNA from RNA. Called transcription, because transcription is making RNA. So the cell makes all of these proteins, whether structural or non-structural, and the tools that are made are used to make more of this RNA, more and more and more. Eventually, the coronavirus is made. One of the interesting things that happened to a cell that is infected with the coronavirus, as I said before, when the coronavirus releases its RNA, the cell doesn't know whether this is a message for itself or it's a virus. It cannot figure it out. It's just an RNA molecule. And it starts making all of these proteins out of it. And it makes the spike proteins. And the cell looks at this spike and says, wow, this is a nice looking spike. I can use it. Probably it's mine. And it starts putting it on its surface, just like the virus would do. The cell starts looking like a virus. This is how our body can figure out if this cell is in fact infected or not. And this is a key component of how the body can figure this out, and really the immune system, which we will probably talk about in a different video, is going to attack this cell because this cell is doing something wrong. It's making the virus. More viruses are going to be released from it at one point, and they will infect more cells and more cells until the person becomes really sick. So the immune system attacks the cell. Now, here's a very nice trick that scientists discovered. How about instead of the virus containing this RNA goes ahead, infects the cell. The cell looks at this RNA, starts making more viruses. These viruses come out of the cell, cause the infection. We just give it the RNA. In fact, we just give it part of the RNA. Which part? As I said before, the RNA of the coronavirus contains regions for structural proteins and non-structural. And the non-structural proteins are very important because they allow to replicate the RNA, and also they contain the spikes. Maybe they're considered structural, doesn't matter. The point is, if I just take the part that makes the spike and not give the cell any of these parts, the cell is not really going to make a virus. There is no virus that's going to be made. It's just going to make some spikes, which are completely harmless. Because as we said, a virus needs the genetic material inside it, which it cannot make, and needs the surrounding protein, which also it cannot make. So it's just going to be making some spikes. But what's the purpose of this? Why would I give the cell the RNA of the spike that the cell is going to make? What happens is that these cells, when I inject them with this spike RNA, the cell is going to make these spikes and put it on its cell, on its surface. There is no virus in the picture. It's just spikes. There's no infection that is going to happen. But from the point of view of the immune system, it's going to look at this cell and says, wait a second, these spikes are unusual. This cell got to be infected because it can't see what is happening inside. So the immune cell is going to destroy this cell, which is okay at this point because there are no viruses that are going to be spreading. So only a few cells are going to be damaged. However, this immune system is going to go to the records and say, we have seen spikes today. These are not good. It's going to memorize this event. It's going to remember it. In the future, when the actual coronavirus infects the cell, or even before, the immune system is going to figure out and catch the virus early on. It's going to catch the virus early on, and it's just going to eat it because it recognizes the spikes. And this is how we get protected from this virus, by giving our body a heads up that these spikes, if you see them, there's something wrong happening. And this is the concept of an RNA vaccine. You inject a small part of the viral genetic material into the person, letting the immune system know what is going to happen in the future, and the immune system is going to be ready to fight the infection very early on. Other viruses are DNA viruses. And DNA viruses are kind of simpler. So we have the DNA virus, gets inside the cell, it injects the genetic material, which is DNA, and what it needs to do is to transcribe this into RNA, which gets translated to protein, just like in any other cell, and also replicate this DNA into more DNA. The cell can do all of this. It's going to assume that this DNA is its own DNA and everyone can deal with it. So they're kind of simple. The difference is that DNA replication and DNA transcription are processes that happen inside the nucleus, which is not very important, just an idea to remember. Another interesting fact about viruses is that, especially RNA viruses, and I'm going to explain why this is particularly more common in RNA viruses, are prone to something called mutations. A mutation is simply a mistake in copying the information sheet. Think about it this way. Go back to the house example. The box flies in, and you start making copies of this box. However, your copying machine is broken. So when you copy this instruction sheet, parts of it are missing or parts of it are wrong. And when you send these viruses out, the other house, your neighbor, is going to read your wrong instruction sheet, and it's going to make a slightly different virus, probably with one extra attachment or one less. Sometimes these mutations are so bad that you can't even read it. You cannot figure out how to make the virus, so you don't make the virus in the first place. But most of the times, these mutations are only small changes in the virus, and it makes it slightly different from one time to the other. Take the coronavirus as an example. The typical coronavirus has these nice spike proteins. Once it gets copied in the cell, sometimes the cell is going to make mistakes reading or copying the RNA. Okay, so this RNA is going to be made, but there will be some mutation. So this one has a mutation. This one has a mutation. If this mutation is affecting the spike protein, the product virus is going to look slightly different. So this virus is going to have a weird looking spike. The other one is going to have another different spike. The problem with changes in the shape of the protein is that the immune system will no longer recognize these weird spikes. So the immune system looks at this and says, yes, that's a spike. We got to fight it. This is a virus. But these weird looking ones are different, and it's not going to fight it. So these mutations allow the virus to change shape and escape the immune system, pretty much like a disguise. So this is the RNA virus that we all know. Okay. And the immune system can attack it. It looks at it. Okay, this is corona. A small mutation happens, and he no longer is recognizable. Who is this guy? This is just a small mutation. Change in the shape, no longer recognized, and it can cause infections. The good thing is that slowly, the immune system is going to figure it out. It's going to say, wait, this guy is also bad. It's not necessary that the immune system will know that both of them are the same person, but it's going to figure out eventually that this guy is also bad, the same as that guy, because technically they are different. Why are RNA viruses more likely to develop mutations than DNA viruses? It's intrinsic to the copying tool. As I said, if the copy machine is broken, there are chances of mutation. So what is the copy machine in RNA viruses? The copy machine is something that you need to build out of the instructions of information instruction. So it's something that the virus tells you how to make. If you make it wrong, it's going to be a bad copy machine. So it's not the greatest quality of a copy machine, but the DNA copying tools are wonderful because these are made by us. They are made by our cells. And they have been doing it for so many times, making our bodies, and they're way more sophisticated. They have error correction systems. They're really advanced and pricey and all of that. So the copy machines of RNA viruses are poor man's copy machine because they are the DIY copy machine that you make at your home, whereas this one is the industrial level copy machine that you buy for thousands. So the DNA copying system is much more sophisticated because we use it, whereas the RNA copy system is very poor because the virus uses it and it's prone to mutations. That's why RNA viruses tend to be changing all the time. Another example of an RNA virus is the influenza virus, the common cold virus, or the flu, the flu is influenza. So the common cold virus is another one. Human immunodeficiency virus, HIV, the virus that causes the disease AIDS, is also an RNA virus. These viruses are nasty because they can change with time. And these mutations are sometimes tricky for the immune system to recognize, whereas the DNA viruses tend to be more stable. This is pretty much what I wanted to talk about regarding the viruses subject. I hope this three-level approach made it easier for us to understand such a tricky subject in a simpler way. Please let me know in the comments your feedback and how you think about these viruses in these videos. And hopefully we meet in a next subject in the future. The subjects that I'm going to cover are pretty much basic biologic subjects like the immune system, probably talk about cancer, and then see what are the more interesting subjects to cover. Thank you very much for listening, and see you.